Good afternoon, everybody. Welcome at this webinar, The Challenges Within Epilepsy Trials in Europe. This webinar is a collaboration of ECET and Julius Clinical. The program of today, we'll do a short introduction of Julius Clinical by myself. My name is Dr. Lieza Exalto. Then Professor Arzimanoglou will talk about the role of the European Reference Network EpiCARE in epilepsy trials. And next, Professor Trinka will talk about the added value of the European collaboration of epilepsy trials, ECET Consortium. And last, Professor French will talk about experiences of the US Epilepsy Consortium. And then we have live Q&A. Just briefly about Julius Clinical. Julius Clinical is an academic CRO. We are a spin-off of the University Medical Center in Utrecht, the Netherlands. And all our work is based on this triangle of networks. We have strong and committed networks of academic leaders and investigators around the world. And today we're going to talk about epilepsy and ECET. We combine this with science. Our scientific leaders have a broad experience on study committees and are part of international networks of thought leaders. And today we have 3 of them. And the third part of the triangle is operations. We provide end-to-end CRO and SMO services in our well-defined therapeutic areas. And I think we can now just continue. To our first speaker. I think there's a little technical problem.
Yeah, okay.
So we will have a music break in between.
Well, I think you can hear me at least, but I don't know why my slides disappeared. But let me try. You can hear me, no?
We hear you.
Great. I found it. Great. Here it is. Okay. So, welcome everybody. Thank you, Lieza, for the introduction. It is a real pleasure for me to share this panel with Eugen and Jackie. This first, I would say, rather introductory talk would be more to give you some ideas of what exactly are the European reference networks in Europe, because we have an audience which is quite large and they may not have all the details of what is the role of the ERNs, and particularly of the one in epilepsy. And of course, as it was said a minute ago, we will go more to the clinical trials topic, which is our main aim today. So first of all, I want to dissipate some misunderstandings. The European reference networks are not research projects, although they are funded by the European Commission as a network within the health plan. They are not legal entities because they combine many hospitals. They are not virtual in the sense that, yes, we discuss patient cases cross borders, you will see it in a minute, but in fact, we represent a number of hospitals and not one hospital or one university. And so from the very first moment of their creation, they were launched in 2017. The concept was that the European reference networks are networks of healthcare providers or healthcare medical teams that are already recognized for their high level of expertise in diagnosis and treatment of a rare or complex disease, working at a national level of expertise and accredited by the respective competent authorities and a board of members of the European Union. So it is quite complex, but it shows how complex is the selection of the centers. This picture is just to impress the public. It's the number of ERNs. In fact, we have 24 that correspond to different areas of disease. These are thematic networks. And as you can see, the epilepsy one is up there on the right corner, but you have also many other fields of disease that are concerned. Now, when you translate the 24 into numbers, today we have more than 500 hospitals involved. Of course, in one hospital, you can have several medical teams for several diseases, which makes it around 2,000 medical teams, or all areas of disease included. And if you take a median of 10 senior doctors per expert medical team, you have approximately 20,000 doctors that are interconnected within this network, plus, of course, the nurses, the other health carers, and young next generation experts that we need to train, which makes it something like 153 Airbuses full of European experts, a quite huge network to manage, I would say. Now, the first raison d'etre of the ERNs is this one: develop optimal cross-border healthcare pathways for patients with rare or complex diseases. And the reason I insist on this, and on the next two slides, is that all the audience needs to keep in mind the difference between one country like the United States and many countries like the European states or countries and so on. And our role is to take care of the patients with rare and complex diseases. This is what 1 million people look like. The problem is that we have around 30 million people having a kind of rare disease in Europe. And on top of it, they speak different languages, because here we are talking about patients that do not necessarily speak English. And this is, I would say, the main reason for the ERNs. The other thing to be kept in mind is what I was saying a minute ago. When we say European Union, which is not all Europe, it's just the European Union, it makes 27 plus healthcare systems, different 27 medical research policies, 27 medical education programs, and this you add the cultures, the languages in Europe, and we need to travel between the two because of the subsidiarity principle, which, as you know, each country remains an independent entity within the European Union. In other words, just to give an example, data sharing between two European countries is not as easy as the word says, because a number of legislations that can be different between the European countries may influence your trial or the collection of data or the patients you can send from one center to another. Now, the innovative part of these ERNs is that for once at least, the governance of, theoretically, the governance is in the hands of the experts. There are 24 coordinators that meet regularly, that discuss the different areas of disease, and they try, I would say, to combine or merge the different actions that in fact cover these aspects that appear on your screen: sharing expertise, developing clinical research, shaping best practices, guidelines, for example, supporting the national networks, and partnership with patient advocates, which is, of course, a regular and important obligation of the network. And just one example, very rapid one, why this system is efficient in Europe. I will take two examples: the COVID pandemic and the war in Ukraine. For EpiCARE, for example, it took less than 6 weeks to have 3 webinars that were diffused largely all over the world about how to handle anti-seizure medications and COVID pandemic. Because the experts were there, the infrastructure was there, and it was quite quick to just connect the centers, see who knows the topic, produce 2 or 3 webinars, and offer them to all our colleagues for free, no matter where they work. Now, going back to EpiCARE, this is the main, I would say, the core group that obtained the creation of a specific reference network for epilepsies. There are others that are related to neurological disorders. One is called ERN for rare neurological disorders. The other one is for neuromuscular disorders. There is one for metabolic diseases with which we collaborate, and there is another one on neurodevelopmental issues, mental retardation, autism, and so on. But this group obtained the creation of a specific epilepsy network, which, because of the transversal nature of the area of the disease we are dealing with, and we obtained gain of cause, and this was created by the European Commission. As I said, in 2017, it was launched. This is where we stand today. We have 50 members, and you can see the distribution between countries. And if you flash the QR code, you can even go on the website in different countries. The network is represented practically everywhere. And of course, even after the Brexit, because following the Brexit, the UK had to leave the administrative part of the network. But of course, our UK colleagues are still fully involved in whatever is patient management, clinical trials, and so on. And also, if ever a center is not opened yet in a country like Ireland or Greece or Switzerland, we still have collaborating partners that we regularly liaise with on all issues of our work. This is the structure of EpiCARE. There is the upper part of the slide, I would say, is the executive committee. Every member of the network, which is now 50 medical teams, has one representative, and there is at least one general assembly per year and 2 calls, which means that there is permanent work for networking and sharing of practices. We have a dedicated research council that was created to follow the different research projects. The network itself cannot do research, but its members, of course, do research because they are the academic hospitals across Europe, and they are the ones who do the clinical trials and other types of research. And then we adapted, I would say, the structure to the epilepsy care needs. As you can see, a number of working groups are dedicated to specific fields of epilepsy care. The structure is not the same in other ERNs because of the disease specificities, but in our case, you know it. Epilepsy means you need to cover not only the clinics, but the genetics, the neuropsychology, the neuropathology, the neuropsychology, the neuroimaging, the neonatal, and so on. So we have specific working groups with members of all our hospitals dealing with those. And also we have a number of networks which are more, I would say, transversal for the production of guidelines, always in collaboration with the scientific societies, registries and data sharing, definition of levels of care for best epilepsy care across Europe, and so on. And the function, in fact, is also adapted to the epilepsy care needs. This is how the network works and how we take care of an epilepsy patient. I would say in every country, the patient first is seen in his local hospital or treating physician. Then he may be addressed to a local epilepsy center, relatively small one usually, and according to the process of diagnosis and or treatment, he can be addressed at a national reference center, which has more facilities, MRI, PET, clinical trials, new drugs, and so on. And if this pathway doesn't reach a conclusion, the reference center in the country can refer the patient to the whole network and ask for a shared opinion between experts. And there are regular calls through a secured platform developed by the European Commission, to which we upload the cases, we invite the panelists, and we share our opinions on best treatments or best diagnosis, and so on. Now, in parallel of this main work, the ERNs are supposed and are expected, and they are audited for this, to foster clinical research, and not only clinical, to foster knowledge generation. I was giving the example of guidelines, but that's not the only thing we do. And of course, to promote education and training, depending upon the needs of European countries. Now, in EpiCARE, we have the huge chance, I would say, to work very closely with the International League Against Epilepsy. I mean, the sorry, the coordinator of EpiCARE before myself was Ellen Cross, who currently is the president of the ELAE, but also close relationships with the European Academy of Neurology or the European Pediatric Neurology Society. And at each country level, we try to liaise with smaller centers and between the network members. So at the end, how do we interfere with clinical trials at the level of EpiCARE? We have experts in the field that, in collaboration with patient advocates, try to identify unmet needs and establish priorities within the epilepsy field. And this is a very recent publication of a project that EpiCARE was included. It was not under the leadership of EpiCARE, but that's not an issue. It was one of the projects that brought together several researchers across Europe, and the publication came out just a few weeks ago. The other level, of course, is have working groups, as I said, focusing on priorities in a very complex disease area. This slide shows part of the story. As you know, in epilepsy, we have at the same time a symptom, the epileptic seizures that can be different, but you have a number of etiologies that can be genetic, that can be lesional, and that they can be of unknown origin, that can be of metabolic, of immune etiology, and so on. For epilepsy surgery, there are a number of procedures that also need to be decided as early as possible for the patients, and the same applies, I would say, to algorithms or best treatments. So all this needs to be reflected, and this is why within EpiCARE 1, we have an EpiCARE Research Council where the members can come to us, propose research projects or clinical trials or investigators-initiated projects. We can give advice. We can ask advice from experts in other hospitals in or out EpiCARE and control all this. And we have a research project manager that guides all this, and there's a regular newsletter coming out of the network every month. The other, of course, point is that we are, I would say, the privileged actors of all the research calls published by the European Commission, and there are a number of collaborations for research projects that can be, for example, transversal between the ERNs, neurological or not, or can be more related to clinical trial support activities, or can be related to academics and so on. And this whole ecosystem works together, and the ERNs are, I would say, in the middle. And then we translate it in other specific working groups. I just gave you one or two examples, and as you can see from the names or the faces of people that you may know, we try to give the leadership to different people in different countries. So there is, of course, one on pre-surgical evaluation and epilepsy surgery, but also on genetic research, as well as in clinical genetics. And there is a genetic platform, for example, that every medical team that wants to collect data for a given disease can upload a proposal, and others can contact them and then take the regular procedure to build up a trial, to build up a study or a publication. And this is what interests us probably most. We have a specific working group on clinical trials and targeted therapies, led by a number of very well-known people, including Emilio Peruca as our advisor, although he moved in Australia. And the role of this group is to identify the priorities and to support special interest groups of members of the network that want to work on N-of-1 trials, for example, on autoimmune diseases, on tuberous sclerosis, work with patients on patient outcomes, and so on. So what we are offering, in fact, is a structured network of experts that can guarantee the quality of the clinical data, the sustainability of the resources, because, for example, the call for the next 4 years was just published until 2027. Data sharing that respects all EU regulations and data protection, and of course, links with the national reference networks, as I mentioned a minute ago. We have a central registry. We are facing today the difficulties I was mentioning at the beginning of legislations, national legislations for data sharing, but at least all members of EpiCARE have today exactly the same template of the same registry applied at the level of the hospital. So at least we can identify the patients of a given disease quite rapidly, even if the issues related to pouring all these data into one registry are not yet solved from a legal point of view. But the sub-registries are all exactly the same between the members of the network. And finally, in fact, how it functions within a country, you have members of the network, the blue ones, related with the smaller hospitals. All these people together are within the same network, which is EpiCARE, networking of individual hospitals, and EpiCARE as a network can link with any other area of disease, of course, at the patient's level as needed. And all these other networks also have a representation at the national level, which closes the circle in a way and allows the functioning of this system. So within this, we thought that it was extremely important to work on the unmet needs for epilepsy clinical trials because of the complexity of the area of the disease and the need to respect at each step, one, the identification of priorities, of course, two, the design of the clinical trials, particularly when they target a given type of epilepsy. We do not believe that the same feasibility questionnaires, for example, can be used for any type of epilepsy. These are the production of a feasibility questionnaire is a medical procedure that needs expertise, and the same applies to the selection of centers, because we can identify for each rare or complex epilepsy or not that a given institution or medical team has the expertise. And this is why ECET was created, to link all these aspects, the smaller hospitals, the members of the ERN, the pharmaceutical companies, the research projects, and so on. And with this in mind, I will stop my talk here and give the floor to the chair, Eugen Trinka of ECET. As you can see, again, ECET involves a number of people that are experts in epilepsy care, and the list that you cannot read on the left are all the hospitals with which we are in regular contact for the development of clinical trials, for the identification of priorities. Thank you very much.
Well, thank you, Alexis. And indeed, I want to open the floor to Professor Trinka. And to all the listeners, you can put questions already in the box for questions. Well, we have questions at the end of all the presentations.
So thank you so much, first of all, for co-organizing this. And thank you, Alexis, for this excellent overview about the enormous network, I have to say, of EpiCARE and the complexity. So I talk a little bit about the problems which we have in epilepsy trials. And I remember that Jackie French said that we have to do something similar, like she organized and ignited this in the USA with the Epilepsy Consortium. And you will hear about that later in the talk. And I think that was the blueprint in order to improve epilepsy trials. So I will talk about the end of epilepsy, success rates of drug development, and the value of innovation and the added value of ECET, which is the final point. So this is by Edmund Husserl about the crisis of European sciences. Well, I would not take it that far, but there is definitely a crisis in epilepsy trials. And when we look at the whole picture, there's an enormous burden of epilepsy, which is the green bar in the middle, but there is a far-fold higher burden of stroke and Alzheimer's disease, but 25% of stroke patients have seizures, and at least 5, if not more, patients have seizures in Alzheimer's disease. So the global picture is a different one than looking at rare epilepsies only, because one thing what we can learn here is about the fact that seizure-free patients can lead a normal life. Those with drug-resistant epilepsy carry a high burden. And what we call rare and complex disease, the word complex refers to drug-resistant patients. So we are there in the network to cover both angles. So when we talk about trials, we are close to evidence-based medicine. It is at the heart of the healthcare for treatment decisions. And evidence-based medicine requires the integration of the best research evidence, what we have. But what is the best research evidence? We always talk about the pyramid of evidence, where retrospective, series, and case observation have the lowest quality, and the majority of evidence, as we say, comes from randomized controlled trials. And these randomized controlled trials are regulatory trials to a large extent. So when we think about this, to improve the quality of regulatory trials together with the FDA and the EMA is one of the major tasks to improve evidence for finding better treatments for epilepsy patients. And again, Jackie French was here one of the drivers in starting a dialogue between the International League Against Epilepsy and the regulatory authorities. And that is a long discussion and dialogue we have with the regulators. So, for example, when we think about the monotherapy trials in Europe, we have this non-inferiority design. The comparator is carbamazepine controlled release. And when we think on all the new substances, they show no difference to controlled release carbamazepine. Does that really help to inform clinicians what is the best treatment for newly diagnosed epilepsy? Further, if we talk about add-on treatments, and this is data from an uncontrolled single-center experience, we know that about 60% of patients are well controlled with monotherapy or first add-on treatment. And this is the same percentage since the early 1980s. Why is that so? There are several reasons, but one of the reasons is maybe because the complexity of epilepsy is not reflected by the trials where we search for new medicines. And we have a lot of new medicines, first, second, and third generation anti-seizure medicines, and the newest drug here is cenobamate. Overall, in the past 35 years, 30 billion US dollar or euro or Swiss francs have been spent for 20 new anti-seizure medicines. And still there is only a handful as successful as the old classical drugs are. So let's look at the problem we have here, and I only select two. The first is the typical add-on design, which is the regulatory trial by FDA and EMA, which still has placebo. So there is a risk for placebo patients to be involved in these trials, which is not negligible. These trials, they don't reflect what you do in the real world. They have a fixed study design, forced titration, a short observation period, which can be a problem if you have a distribution of seizures like you have here. Let's say baseline monthly seizure frequency is about 4, and you see the distribution of those 4 seizures. One patient has them all in week 3. So then this patient may enter a seizure-free period, and you will wrongly associate that with the treatment. So this is one problem of regression to the mean. The second major problem is how reliable are seizure counts. At the moment, we rely on seizure diaries. Well, all these CROs and Julius Clinical, forgive me, we look at the seizure diaries, whether the patient put that in. But how on earth can the patient do that when the patient doesn't recognize his or her own seizures? And you see in this work by Mark Cook that there is no correlation between the reported seizure frequency and the recorded seizure frequency here in an intracranial implantable device. That is maybe also the cause why trials fail in neurology and especially in epilepsy. When we look at the preclinical phase 1, 2, 3 until the regulatory success rate, until market approval, the chance of progressing overall is low and the costs are enormous. So this graph from Wolfgang Löscher is 10 years old, and the total cost for bringing one drug into the market is probably 10 times higher as it is here now. 2 to 3 billion is the effective cost. The time is still the same. It's about 10 years. So why should industry invest in trials when the success rate is not high enough? And the overall success rate, as I said, is low, especially in neurology. I come to that point. But there is a transition phase from phase 2 to phase 3 where the probability of success is very low. It's only 1 out of 3 drugs which are successful in this phase. And phase 2 and phase 3 trials are in the clinical arena. So this is something where we can have an influence on that. Let's look at neurology trials, and you see the success rate from phase 2 to phase 3 is about what it is overall, but the overall success rate of neurology trials is far lower than the average indications. And this is what you see here, probability of phase 2 success on the left and probability of phase 3 success on the right. Let's look at neurology. When we pass phase 2, it's even worse and below average, the success rate when we come to phase 3 trials. In other indications, it is completely the opposite. For example, in urology, once urology passes phase 2, it's very successful in phase 3. But neurology does equally bad. So what can be made here? And one thing is biomarkers. When we introduce biomarkers in that field, pre-selection and enriched populations, you see the transition from phase 2 to phase 3 is much better with biomarkers, and the overall improvement from phase 1 to approval, the overall success rate almost doubles. And this is something which is important. So biomarker are one point which I want to mention, improving both clinical trials, phase 2 and phase 3. We are dealing with rare diseases, rare epilepsies, and if we look at the rare disease indications, again, the success rate here in red, as opposed to the chronic high prevalence diseases, let's say, there's nothing like a standard epilepsy, but the common epilepsies with no further specification is twice as high from phase 2 to 3, and almost 3 times as high as when we look at the overall picture. So rare disease is an incentive. But what is epilepsy? Well, we may say this is the end of epilepsy, and Alexis Arzimanoglou said it. They have one common denominator, and these are seizures. So if we think about all the diseases which make seizures, well, we have nothing like one uniform epilepsy. So the concept still in the regulatory protocols is probably outdated. But when we look at epilepsy, we have different causes, as you see here, from mitochondrial to tuberous sclerosis, to low-grade tumors, to genetic imprint. We still put everything in one basket with randomized controlled trials. We stratified mostly according to the seizure type in monotherapy and in add-on in refractory. First of all, why is that so? Well, if a drug works in monotherapy, it's also very likely that it works in combination treatment as add-on. And the other barriers which we have in randomized controlled trials is the randomness of seizures, the regression to the mean as indicated previously, the inability of patients to recognize seizures, and the small effect size in newly diagnosed and refractory patients. So this is why when we know something about the epilepsy, for example, the mutation and the course of the disease, the natural history, we can deal also with lower numbers. And this has been incentivized by the regulatory authorities. But still, we have a problem. Epilepsies are dangerous group of disorders and they can kill you. So the rate of sudden unexpected death in epilepsy is high. So 1 to 3 generalized tonic-clonic seizures per year tells you that the patient has a 10-fold risk of having SUDEP or a 19-fold risk of dying with SUDEP when the patient is below 16 years. So when we do trials in epilepsy, we have to acknowledge that placebo is something very problematic. So coming back to the whole picture, our rare genetic disorders is one group of patients where we have to develop trial designs and perform trials in high-class centers as we have it in the EpiCARE. And the other ones, the so-called common epilepsies, they can profit, the high prevalence epilepsies can profit from the experience there. So putting together the industry perspective, here we have it as summary. Rare disease therapies are incentivized. The likelihood of approval overall is low in the indications we treat with. The biological complexity is enormous in epilepsy, and it is also in other diseases, and you have here examples of CAR-T treatment and RNA interference, which achieve higher likelihood of success. The biomarkers are important, and the transition from phase 2 to phase 3 is critical for the development. So there is expedited pathways when the benefit risks ratio can be assessed positively, where there is an unmet need and where there is a major interest from public health perspective. And expedited pathways from the EMA, they have been mostly using surrogate endpoints or biomarkers. So when we think about that and we think about trials in Europe, we need centers which can fulfill these needs. So let's say one biomarker in epilepsy, it's very likely that EEG is the best biomarker which we have. It predicts recurrence of seizures after first unprovoked seizures. It can provide important information that helps to classify epilepsy, but it is also a source of misdiagnosis, misinterpretation, and overreading of EEG, or underreading of EEG, underreporting, is an enormous problem. So you need centers which are here extremely experienced, reliable, and this is key for the inclusion criteria. So we have developed standardized terminology for intensive care, standardized EEG reporting under the leadership of Shandor Binitsky, and also automated tools are here available, and that runs within the centers. The second important point, as I mentioned previously, is the development of long recording EEGs, which can help improving the trials. And these techniques are only available in high expertise centers. EEG can also be used as, as a surrogate marker for proof of principle trials. So we looked at, for example, at Montelukast in a pharmaco-EEG study in a cognitive task in only 12 patients with asthma, and we found significant changes in the entropy, which decreased, and an accelerated background rhythm. So this was a proof of principle that Montelukast passes the blood-brain barrier and has a CNS effect with this drug. Low number of patients, significant result. So why do we need ECET? European Collaboration for Epilepsy Trial, and I said the blueprint was the US Epilepsy Consortium. So a network of specialized epilepsy center for the conduction of high-class trials, to facilitate the collaboration with the patient groups according to the content and aim of the trial, to implement educational activities to improve the expertise of the researchers and healthcare personnel involved in the trials, and to provide professional expertise in the design and implementation of clinical trials adapted to the field of epilepsies, to give advice for industry and for academia. We also want to promote and facilitate academic trials in the same way as we want to improve regulatory and industry trials. And we also provide advice and professional expertise as per protocol on the eligibility and related task when industry wants to perform a trial in Europe. but we are not a CRO, as we very often mention. We are a consortium, a collaboration of different entities, more than 47 experienced high-class clinical trials across the European Union, UK, Norway, and Switzerland, and also outside of Switzerland, for example, in the Eastern Europe for selected trials. So there is high expertise in drug-resistant epilepsies, in rare genetic epilepsies, mainly within the EpiCARE network, with status epilepticus, adults, children, and elderly population. We are experienced in using biomarkers, mainly clinical neurophysiological, but also neuroimaging biomarkers, and in the performance of anti-epileptogenesis trials. And one of that is going to be published in the next few weeks, a proof of principle trial for anti-epileptogenesis. And there is a close link to EpiCARE and to e-epilepsy, which has been the prototypic European Reference Network for drug-resistant epilepsy and surgical epilepsy treatment. So what can we offer? Our mission is to improve the clinical trials in epilepsies in Europe, and we aim to ensure consistency and reproducibility, and thus the reliability of final results of clinical trials. Advice on clinical trial design to increase the success rate from phase 2 to 3 for industry and for academia. Selection of trial sites in Europe and beyond. This is something usually CROs do, but I think we can do it better because we know the trials and we are not moving from one field to the other. And one center may be excellent in Parkinson's disease, but may be extremely poor in epilepsy. So this selection is a critical point. And the group of advisors for clinical drug development in all phases. At the same, we advise academia or small startup companies. And one thing, as a service, we can offer centralized and standardized adjudication process, looking at the inclusion criteria, looking at the EEG, looking at the endpoints to reduce the variability in multi-center and in multinational trials with 40 different languages, 40 different cultural backgrounds of the doctors. And to advise in biomarkers for enriched populations, including anti-epileptogenesis trials. And there is, of course, also expertise and experience in trials with low numbers. How do we work? We have a project manager, Sibyl Cajca, and she's on the homepage, and you saw her contact point already, or any member of the steering committee. We have regular meetings of the steering committee at all major conferences, and all EpiCARE meetings at least twice a year. So there's a close contact on a continuous basis. And we are gratefully indebted to the US Epilepsy Trial Consortium. Jacqueline French gave us a large amount of advices, how to set up this, how to run this. And we have an informal experience exchange on a regular basis. We can discuss the project someone wants from us. We agree on the package from simple advisory committees to the full scientific advice for a clinical trial. Then there is a contract with ECET, and you can get started whatever you want to do. So to conclude, epilepsies are different than most of the other neurological disorders. It's a heterogeneous group of disorders associated with a high burden of disease. Seizures are the key symptom, but they are not the only one. They are among others. Clinical trials in epilepsies are therefore complex and demanding, and there's a need for highly experienced collaborating centers to perform these. We are very much interested in innovative trial designs and biomarkers for enriched populations and trials with low numbers. And we feel that ECET forms an ideal hub for industry and academia to perform these trials. And I thank you very much for your attention.
Eugen, thank you very much for this nice overview. As a neurologist, I have to say I'm a bit underwhelmed that since the 1980s, we haven't made any progress, and that as neurologists, the field of neurology does so bad with progression of new drugs.
So let's do better trials.
Yes, let's do that. Professor French, I'm very happy to introduce you as the last speaker, because apparently, as Eugen told us, you have prepared the blueprint for ECET. And we're happy to learn from your experience. So the floor is yours.
Thank you. Can you just project my slides, please? And I think I'm going to echo a few things that Professor Trinka said while I provide you with the experiences of the U.S. Epilepsy Study Consortium. Are you seeing the slides in full PowerPoint mode? Okay.
Yeah, that's fine.
All right, go to the next slide. Because I work with many companies in this space, can you go to the next slide, please?
Yeah, I think you're not yet at plenary mode. That's why they don't move, because we can see the slides on the left side.
They should be being presented by somebody. What do you see now? Yes, so can you put it in presenter mode, in slideshow mode? There we go, and go back one slide, please. There we go. Sorry, the one in the middle. There we go. So these are my disclosures because this is a successful consortium working that we are working with all of these companies with the Epilepsy Study Consortium, which is a nonprofit, to speed new therapies to people who need them. Next slide, please. So the mission statement of the Epilepsy Study Consortium is that it is a nonprofit and it is a group of scientific investigators from academic medical research centers who are dedicated to accelerating the development of new therapies in epilepsy to improve patient care. And the goals are to build a partnership between academics, industry, and regulatory agencies, and optimize clinical trial methodology in order to responsibly speed new treatments to patients. So you can see that there are a lot of aligned goals with ECET. Next slide. So I'm going to talk a little bit during my presentation about effect sizes in trials, which directly relates to the ability of the trial to come home successfully and ultimately get the drug on the market. So clinical efficacy is determined in placebo-controlled trials by effect size, and the effect size measures the impact of the drug that's theoretically due to the drug's action alone after excluding other factors. And we think about the placebo responder rate as being a main factor, but there are other things going on, including noisiness in the trial, variability in the trial, issues with the trial conduct. And the effect size is measured as the percentage of subjects improving on the drug when you subtract the effect size in people who are not on the drug. So they are in the placebo group. That doesn't mean that everything that happens in that group is traditional placebo effect, as we'll see. Next slide. So here is an example where you see the results of a lacosamide trial that had 3 arms. And you can see that in the low, middle, and high dose arm, there was a nice, robust 50% responder rate in all 3 arms. However, if you go to the next slide, you can see that the placebo group actually had a 22% response. So when you subtract that, you actually only get a meager response, particularly from the low dose group of only 11%. So obviously, if the placebo rate had been lower, we could attribute more of that effect to the actual drug. Next slide. So how can the effect size change? Issues with the conduct of a trial can do several things. The placebo rate can go up, the treatment effect can actually go down, and in either case, that would shrink the actual effect size. And at some point, this may, and in fact has, led to failed trials due to lack of differentiation between the active and placebo arms. And I would even point out, and I think this point was kind of made with Professor Trinka's slides, that when you see no difference between drug and two drugs in newly diagnosed studies, you don't really know what the placebo or noise effect is. So you don't know whether you are masking a difference between those drugs. Next slide. And in fact, effect sizes have been going down over time. And this is just from 1990 to 2010. They have gone down even more in more recent times. Next slide. So the question is, why are effect sizes going down, and what can we do about it? Several recent trials have indicated that changes in effect size actually can arise in noisy subsets of patients within a trial. And again, this goes back to Professor Trinka's point about selecting the right centers to do studies. Understanding and identifying those subsets really helps us understand why our effect size may be disappearing. Next slide. So here is one very cogent example. I could give you many, but in the interest of time, this was the results of the perampanel trial. The pale green is placebo, and then there's 2 doses of perampanel. In North America, you can see that the placebo responder rate was relatively low at 10%. The medium dose was reasonably good at about 30%, and the high dose was even better, close to 40%. Half of the study was done in North America, almost half, and the other half was done outside of North America. In this case, it was actually some sites in South America. And lo and behold, you can see that in South America, the placebo responder rate was the highest response of all the groups. There was no dose response relationship. And if you had done the entire study in South America, you would have lost the ability to determine that this drug, in fact, had any effect at all. The interesting thing to note is that the study would have been statistically significant at both doses with far fewer patients if it had been done in North America alone. And believe me, I'm not saying that all sites in North America have less noise, but in this case, you can see that the noisiness in the study was far lower in North America than it was in South America. Next slide. So unfortunately, in 2023, smaller centers, private clinics without as much experience are responsible for most of the patient enrollment in therapeutic trials. And that means that inappropriate seizure classification can occur. Patients without epilepsy can be enrolled. The physicians don't necessarily have a dedicated study staff or as much time to evaluate patients. And in some areas, diagnostic tools are less readily available, such as video EEG, MRI, and expert EEG reading. And we already heard about misinterpretation of EEG. Next slide. So when we saw these things begin to happen, the study consortium jumped in to try and salvage at least some trials and some outcomes and improve the effect size and perhaps change the direction of that curve. So first of all, we did investigator training on how to interview and properly classify subjects. And we developed and conduct an interactive seizure classification quiz for investigators that they have to pass in order to enroll patients in the trial. In addition, we added adjudication or central review of clinical trial participants to try and get a remote perspective on whether they really belonged in the trial, whether they were properly selected, and whether the seizures, in fact, were classified correctly and consistently across sites. So we created a seizure identification form, and that needs to be submitted by sites during the screening phase. That is when they're in baseline. That means it doesn't hold up the enrollment of the subject, and it needs to be approved before the subject is randomized. These forms are reviewed by clinical reviewers who are Epilepsy Study Consortium members or consultants who do have experience and expertise in the conduct of clinical studies within this disease area. So you're bringing expertise in where there may be less expertise. Next slide. There we go. So this is what the form looks like. It's pretty straightforward. We get a detailed description of the seizure, ask how long it lasts, whether discrete and isolated seizures can be counted, which of course is very important in terms of outcomes of trials, counting of the events. We ask if there's impaired awareness, whether there's a warning, and if so, to describe it. And then we ask the investigator to tell us what they would have classified that seizure as if there was no remote review. Next slide. So when we started doing this, we realized immediately that things were not all, daisies and flowers. So this is an early form. Again, we asked for a detailed description. And this description here is that the seizures start as a rushing, burning feeling in her thighs and abdomen, then nausea, then dry heaves, all lasting about 30 seconds without loss of consciousness. And all seizures are this type. And there are a couple of problems here. The first is that in the absence of an EEG abnormality, I think all of us would wonder whether a rushing, burning feeling in the thighs was actually a seizure manifestation. But the second thing is that they clearly did not understand seizure classification because they clearly say there is no loss of consciousness. And then they call this, in the old parlance, a complex partial seizure, which is a seizure type that in fact does have loss of consciousness. So this is exactly the type of classification that the study consortium can jump in and say, Houston, there's a problem here. Next slide. But we noticed that there were problems above and beyond just the kind of problem I showed you. So this is a little difficult to read, but at the bottom, I've typed out what this investigator said. Without warning, the patient loses awareness, wakes up in 1 to 2 minutes. The family reports he will get a glassy-eyed stare. He babbles unintelligibly and has chewing movements, complex partial seizure. So on the face of it, this looks like a beautiful description of what we now call a focal impaired aware or complex partial seizure. So what was the problem? Next slide. This is a second form from a different center where the seizure is described as the patient with no warning is reported to have chewing movements. The family reports he will get a glassy-eyed stare and babbles unintelligibly. So now we are saying, two centers, not the same center, same description, almost word for word. This was quite suspicious, and this allows us to identify suspicious activity within a trial. Next slide. And then we went back and looked at the seizure quiz question that we had provided to the investigator. And of course, in the seizure quiz question, the individual gets a glassy-eyed stare and babbles unintelligibly. So what's going on here? It's interesting that these were two sites that were in the same geographic region of a single country, but two different sites. So what's going on? Are they talking to each other? Do they not understand how to do the process? Are these real patients? There's all kinds of questions that come up in your mind. So this allows a sponsor to go and try and dig into what's going on in this particular circumstance. We've also had circumstances where the seizure description doesn't come from the quiz, but for example, a center will enroll 7 consecutive patients and all of them will have almost identical seizure descriptions, which of course we know does not actually happen in the real world. So asking for people to describe the seizures that are occurring and having remote review allows us to at least put a little bit of a damper on the noise that we saw creeping into trials. Next slide. So the form has evolved, and whereas the seizure identification form, as you can see, was quite informative, it didn't really allow us to determine the likelihood that the patient actually had epilepsy. So once we started to see the results that we got, we added a diagnostic review form that actually asks for neuroimaging, EEG, etiology, current and past seizure types, developmental history, etc. And these are really useful in allowing us to determine the likelihood, for example, in that first patient, that they actually do have epilepsy. And, you know, our rule of thumb is that we have to have an 80% certainty once we review all the information that not only the patient has epilepsy, but that their epilepsy has been appropriately classified to allow them to enroll in that particular study, whatever the study is requiring. And you would think that this is, for example, not important for the genetic epilepsies because they have to have an abnormal gene, but we have found that even in those cases, there's about a 5% misclassification of patients being enrolled in a trial. Next slide. So today we've conducted seizure identification and diagnostic review forms since 2010 for more than 40 sponsors, more than 90 protocols, 45 investigational products, 30 indications, types of indications, and 9,500 participants. My people work pretty hard. We also provide other services to facilitate anti-seizure medicine and hopefully anti-epilepsy medicine in the future, therapy development. We assist sponsors with design and conduct of clinical trials. We help them with development of the protocol and the case report forms and source material. We do photosensitivity studies as proof of concept. We can rapidly generate advisory boards when necessary to address some thorny issue that comes up or assemble a DSMB. We do central EEG review. We have had an epilepsy therapy and device development meeting every other year since 1992. Since the coordinators are really the heart of any study, we do coordinator boot camps. And we also provide grants for epilepsy fellows for people who want to assess epilepsy therapy. Next slide, and last slide, I believe. And I just want to let anybody know who's interested that our next Epilepsy Therapy and Diagnostic Development Symposium, which used to be the AEDD meeting, is now the ETDD meeting to highlight that we don't use the word anti-epileptic drug anymore. Will occur May 31st to June 2nd, 2023, and it is hybrid. So here is the link, and we hope some of you will be interested in joining us. Thank you very much.
Jacqueline, thank you so much for this wonderful presentation. And I want to thank again Alexis and Eugen also for this very nice overview. I think it was a very nice build up with the ARN and then the ECET and then the examples from the US. We also have some questions. And I think there's one very interesting one, and I think it's for all of you. If we were to use ASC in the US and ECET in the EU for a trial, would the results be sufficiently comparable? Is a question by Javier Avendaño. I'm sorry if I mispronounce your name.
So one thing I wanted to say to start out is that we perform somewhat different functions, right? Certainly, we don't do the trial. We're not a CRO. So, you know, if you use two different CROs, that's one thing. But the trial is going to be conducted the way the trial is going to be conducted. We have actually had conversations with ECET. We have a very robust process in place for adjudication of subjects, but most of our reviewers are English speaking. So we are working with ECET to have them incorporated into the study consortium to review in the language of the investigator. So they would still use the same process, but they would be doing it on behalf of ECET, and they would be able to provide additional languages. So I don't think that that would make a difference. And as far as who knows where the best study centers are, I think we can say that the people in the region are probably better capable of determining study centers. And I'll let Eugen and Alexis chime in.
Well, I agree with you, and that's a very important point. So the cultural, the language heterogeneity of Europe was a major obstacle, and the way to overcome this is with networks. So I think the proof of principle, Lieza, would be to compare the results in the same study with the same CRO. Very often it's a different CRO in USA and in Europe or in other countries of the world. It's not necessarily the same, but using the same good CRO, there are not too many about that, and to compare the results. And I think the previous results, as you showed, Jackie, to say that in the USA there was a low heterogeneity, there was a highly significant results, and all the other centers and the rest of the world, I have to say, introduced so much noise that the study almost failed. And this should not be the case. So if we do that ever again, I would be happy to see that the quality really improved to a point where we can lower the overall numbers of patients to be exposed to a study drug, right?
No, I have nothing specific to add. I think Eugen and Jackie covered the topic. I'm just insisting on what was said, the importance of the selection of centers that know the specific epilepsy disease that corresponds to a trial for all of us. This is essential. And probably most of the mistakes come from there. A genetic epilepsy is not treated the same way with a surgical one and so on. And you have medical teams that have all the expertise. You have others that have less, but they need to be selected appropriately by those who know what the centers are publishing and what they are producing in the past 10, 20, 30 years. And that is a crucial point. And it applies also to the feasibility questionnaires, as I said a minute ago. I mean, many of them start, I keep giving this example because it's so shocking. Many of them start with the first phrase, are you experienced in epilepsy? We decided you are an expert, can you answer? I mean, this is just 5 minutes of PubMed to find out if someone is interested in epilepsy or not. And it shows that it's not adapted to the content. So I think it is important. And as for the difference between the two, you see very well that we work together all the time. So I don't think it will be any difference between.
Well, thank you for these answers. And as I'm from a CRO, I'm happy to take on the challenge, Eugen, that you propose. Let's continue.
I just want to add one more thing. I'm working on a paper with a colleague of mine that shows that not only have effect sizes gone down over time, but the number of patients that need to be enrolled in trials continues to rise over time, and it's very clear from the data. And it's a vicious cycle because the more subjects you need, the more places you need to go to that are not those expert centers. I wish we could only go to those expert centers, Alexis, but there aren't enough of them to enroll enough patients to perform trials in 2023. We can talk about why each center can only, enroll a small number of subjects. And so there has to be a way of also supervising the centers that, at least as of now, have to be used and are less experienced, as well as making sure that every experienced center has an opportunity to contribute to the trial.
No, I agree. And And that is why I stressed the role of networks such as EpiCARE or what the consortium. This is the whole difference. If you have smaller centers with less experience, it is completely different if they are involved in a trial under the hat of a network where they have support, they can bring in patients, than just saying, particularly in Europe where we have many countries, just saying to one center, can you ask your colleagues to send you the patients? It doesn't work that way for several reasons. But it works if a network with knowledge of the big and the small centers links all these people together and feeds the trial and the number of patients they need. That's...
Agree.
I'm also having an eye on the chat because there's some questions there. There's some worries about sending data overseas, but there's great trust in both consortia, so that's good news. Danielle McDermott says, given the concerns raised, should there be an education process for centers? This has many downstream consequences, not only the potential to affect drug approval, but more concerning patient safety concerns.
The answer is yes. And you heard the boot camp, well, that's education. And we have educational seminars very closely linked to the guideline work package in the EpiCARE. So education is really not only helping improving the trial quality, but as you said, it's about the patient safety. To recognize adverse events early, which may be completely new. Think about these patients who turned blue when they took thiagabine. And think about the visual defects which they had with vigabatrin. It was discovered too late. So new anti-seizure medicine may have adverse effects which you have to observe keen. It's about patient safety. And the other one is reducing the exposure to placebo in this high seizure frequency group in addition. So that's important. And that can only be with information and observation and careful recording of any nocturnal seizures, for example, or assessing all the environments, whether the patient sleeps alone or not, etc.
I see that Conrad had an early question that didn't get asked yet.
Yeah, feel free to, Jacqueline.
So he said, would there need to be adjudication by disease experts rather than just experts in epilepsy in view of trials and more specific syndromes? And Conrad, I completely agree with you. Whenever we set up adjudication for the study consortium, we have that very much in mind. So for example, when we adjudicated Dravet, we set up a specific review panel that consisted of the top Dravet experts in the world. Not all in the US, for example, Professor Ingrid Scheffer and professor and at UCSF. So we do very much keep in mind who are the correct people to adjudicate. And I personally would not ever adjudicate a Dravet study because I would be very bad at it. So you have to be specific.
Thank you, Jacqueline. There's also some questions in the Q&A part. Tina Djukic, she was very early with this question already, and full disclosure, she's from Lifeline's Neuro Company, an equipment service. And she's asking, with EEG, the consistency of the equipment in different countries can also be a barrier. How do you address this?
Yes, it can be, but there are two very important movements and achievements in the last years. First of all, it's DICOM. It's a standardized format which you can use and analyze the EEG, for example, centralized, but you have to record it appropriately with a regular machine 1020, which is compatible. If devices are used, the problem of heterogeneity can be quite strong. So there, if you use an EEG device for rapid recording, for example, it has to be the same in each center. Otherwise, there can be too much noise. But for the regular 1020 recording, I think DICOM format, standardized reading, and standardized terminology score, for example, reduces the misdiagnosis, and there's a very high kappa already in EEG expert reading in many countries.
Certainly not in every country.
Certainly not in every country.
When we review, it's not only the equipment, it's the montages and the EEG interpretation, of course.
Yeah.
And that really varies from country to country. When possible for our diagnostic review, we actually get a screenshot. And it's like fascinating, for example, how many countries will, to get a little specific, when you're doing a generalized epilepsy study, they'll give you only reference mode shots so that every epileptiform abnormality shows up in every channel. That's a good example of, how it can change by montage.
Then at least they know something about EEG in order to make up a generalized recording. But you see, that's also used for publication. There is a publication where they used simply this. But I agree absolutely with you. And there was a shocking paper in JAMA Neurology showing that the kappa value in a high-class, highly renowned North American center, I don't tell you which one, but you can look it up in the PubMed, is 30%. 30% agreement. So there's really, this is why we have definition. This is why we have minimum standards. But if we don't care about education, implementation of these standards into the centers, we will not achieve anything, because that's nice publications, but we have to bring it to the ground. That's important.
And then, I agree that the consistency of the EEG material is an important question. It may influence, but I am convinced that the quality of the reading is more important. And I would say the reading in both sides, both, of course, of a consortium that controls what comes in, but also of the site reading. And this needs to be adapted to the trials also. Because I remember a trial in my department, which is a neurophysiology department, where because the equipment came from outside, qualified epilepsy nurses would not follow what was going on during the recording because it was an external machine that it was not adapted to. So they were completely lost, and the expertise of the center couldn't help the study because we couldn't see the EEG while they are trained to look at them all the time and test the patients and so on. So that's why I keep saying they need to be adapted to the trial and to the needs and to the centers, and it is not one fits it all. This doesn't work.
I also will say that there's a role of the sponsor because for certain studies, the consortium may only have access to what the clinician says the EEG says, for example, which is really not helpful at all, and then we may only have access to the report, which in some cases may not be helpful. And then, obviously the best of all possible worlds is when we actually have access to the EEG abnormality itself in a screenshot.
So I now want to go to the last question from the listeners. This is by Karen Gammon. This is for Jackie. Thank you for this excellent presentation. In the US, FDA guidance for diversity plan became law. Understanding the benefits of major centers, do you have insights on how to meet the requirement to include underrepresented racial and ethnic groups?
I do, but it is not going to be easy, I'm sorry to say, because the centers where those people are seen, that's number one, are not usually the highly expert centers. So I'm going to give you an example. At my institution, we have NYU Epilepsy Center, and right next door we have Bellevue, which is a city hospital. That's where many of the underrepresented minorities are seen for their epilepsy. They have no experience in performing clinical trials. They have no coordinator. So that's issue number one. Issue number two is that The seizures that are experienced are often, driven by things other than whether the medication is working or not. There's a certain amount of chaos and crisis and picking up your medication and not being able to, or having something else fall through. So getting those sites to pick the appropriate patients and to be able to do those studies is going to take a heavy lift that obviously now, I think it's reasonable and appropriate, but the pharma companies are going to have to collaborate with us in lifting those centers to the point where they are in a position to enroll patients. Because I can tell you, it's exactly what Eugen said, or I can't remember, it was either Alexia or Eugen, to tell Bellevue, please send your patients over to us to enroll in trials, really doesn't work. You have to have people that look like them, that have given them regular care, that are involved in those trials.
Yes, I totally agree. And I also think that this challenge doesn't only apply for epilepsy trials. It concerns all major indications. And I think there's big challenges there as well. I want to close this webinar by asking all three of you to answer in 1 or 2 lines, because the topic was challenges in epilepsy trials. If you have like one solution, like for one of the challenges you've debated, like, can you just end the webinar with like a positive vibe with one solution you think is very important, but short? Who wants to go first?
All right, I'll kick it off. I think that if the experience centers are able to enroll more patients, we will reduce the number of centers we need, and we will reduce the noise. So it is critical for us to increase the number of subjects that can be enrolled. And our solution that we're looking very closely into is remote visits, so that the subject can be enrolled without having to come so frequently to the center. And we think that that will definitely increase our ability to recruit.
Very nice. Thank you.
I fully support that. So there's one solution. And the other one is regarding the process. I think the process, when industry wants to conduct a trial, it's very important to consult early. A network which is experienced in doing that, giving advice. And then the entry point is the network in the US, is in Europe. And then from there, CROs are involved, but not the other way around. If the process starts from the backside and from the CRO, I think it will not work in the same way. So that's my suggestion for improvement.
Yep. Well, that's why I waited for the two of them. I don't have much to add to that. I think that's the most important point is, again, the quality of the selection of the patients into the trials, which is unavoidably and immediately linked to the quality of the selection of the centers and, of course, of the design of the trial. But all this needs to start very early with those that have really knowledge on how to develop clinical trials in adults, children, neonates. I mean, the settings are not the same and cannot be reproduced. There are categories in epilepsy, but they are just categories. We don't need to go per disease to find the centers, but we cannot mix all types of epilepsies as if it was the same, unless these are huge centers that have a lot of experience with the necessary clinical staff, with the necessary research assistants and so on. Otherwise, it rapidly becomes extremely difficult to catch up, and it leads to an exponential increase of centers, which increases noise and increases results that cannot be interpreted. And with a lot of money spent on top of it behind it, that's goes together.
Well, again, I want to thank Alexis, Eugen, and Jacqueline for your wonderful presentations and the nice debate. There are clearly huge challenges for good clinical trials in epilepsy, but we also have debated some possible solutions, and especially the power of good networks with experts that can lead the way. So I want to thank you again, and have a nice evening or afternoon for who's are not on European time.
Well, I would like also to thank Julius for this initiative, because I think it shows how important it is to establish a regular collaboration and thinking in advance about how this can be developed. As Eugen said, the CROs afterwards play a very important role in these trials. We are not CROs and we will not do it. But the collaboration is extremely important from onset if we want this to be successful. So thank you again.
Thank you.